Hello everyone, welcome back. Today we will discuss one important topic in cell biology, that is cell cycle. In this lecture we will discuss about the cell cycle, its introduction, then we will discuss about the mitotic division. So let's start. Cell cycle. All the active cells or metabolically active cells which have not been differentiated, जो कि differentiate नहीं हुए हैं, they are generally destined to divide, means they will divide and will multiply themselves और उनका number increase होगा, finally they will differentiate to perform a particular function. So what is cell cycle? It is a duration from one cell division to the next one, और जो भी इस time period के बीच में जो भी events आते हैं they will form the cell cycle. So it is the duration from one cell division to the next one is known as cell cycle, and the cell cycle of eukaryotic cell is divided into two fundamental parts which consist of interphase and mitotic phase. This interphase is the longest duration. In this phase, cell will prepare itself for cell division, और mitotic जो division है M phase, इसमें cell का जो nucleus है that will divide, followed by division of cytoplasm, that is cytokinesis. So this interphase consists of further different subphases. First one is the G1 phase, which is known as gap 1 phase. In this phase, cell prepare itself, it is metabolically active and increases in size, और इसमें जो RNA है proteins की synthesis increase हो जाती है and prepare the enzymes which are required for DNA replication, and after that it will enter into the next phase, that is S phase. और जो S phase है इसमें होता है DNA replication, और DNA का जो amount है that gets doubled in S phase, and after this there is also formation of histone proteins. जो DNA के साथ इसमें histone proteins भी बनती है. Then it will enter into the G2 phase, that is gap 2 phase or growth 2 phase. यहाँ पे remaining proteins और जो भी requirement है cell division के लिए वो होती है, and the cell enlarges in size, maximum size attain करता है, and after that it will enter into the M phase, that is mitotic phase. It consists of two parts. One is karyokinesis, that is division of nucleus, and second one is the cytokinesis, that is division of cytoplasm. So this is the cell cycle. और इसमें जो तीन phases, सब phases हैं interphases के, like we have G1 phase, then S phase, G2 phase. So these three phases are combined to form one phase, that is the interphase. Then after interphase, the cell will enter into the mitotic phase, that is M phase. It consists of nuclear division, that is karyokinesis, and next one is the division of cytoplasm, that is cytokinesis. Then it will give rise to the two daughter cells from one cells, which are exactly same in case of mitotic division. इसमें chromosome number same रहेगा in case of mitotic division. So this is the brief idea about cell cycle. In next slide there is one animation that will make you clear about the cell cycle and mitotic division.
An adult human contains an estimated 100 trillion cells, and yet we start life as a single cell. To grow, develop, and repair tissue damage, we rely on cell division. In eukaryotic cells, this process is accomplished by a series of well-orchestrated steps called mitosis. Every day our bodies must produce millions of skin cells to replace those lost through normal activity. Each of these cells must have a complete complement of the genetic material. Prior to cell division, where the cell divides into two identical cells, the DNA needs to be replicated so that each daughter cell receives an exact copy. Following DNA replication, the chromosomes condense in the nucleus of the cell. DNA condenses by wrapping around cores of histone proteins, forming nucleosomes. This beads on a string structure is called chromatin. As a cell prepares to divide, chromatin coils up further, shortening and condensing the chromosome. The replicated chromosomes are called sister chromatids. The replication of DNA and the formation of sister chromatids is one part of the entire cell cycle. To prepare for cell division, the cell goes through interphase, which can be divided into three distinct phases. During G1 or gap 1 phase, all the organelles and cytoplasmic components, including the centrioles in animal cells, replicate. Then during S, or synthesis phase, the DNA replicates. Finally, during G2, or gap2 phase, all the enzymes needed to aid in the process of cell division are produced. Most eukaryotic cells spend a great deal of time in interphase, and a very short period of time actually dividing, a process called mitosis. The cell is now ready to go through mitosis, which consists of prophase, metaphase, anaphase, and telophase. During prophase, the chromosomes condense and become visible, appearing as two sister chromatids held together at the centromere. The cytoskeleton disassembles as the spindle begins to form. In animal cells, centrioles play an important role in the distribution of the chromosomes in the dividing cell. The centrioles migrate to opposite poles, establishing a bridge of microtubules called the spindle apparatus, and the nuclear envelope breaks down. Towards the end of prophase, chromosomes attach, by proteins in their centromeres called kinetochores, to microtubules from each pole, moving the chromosomes toward the equator of the cell. During metaphase, all chromosomes are aligned at the equator of the cell, called the metaphase plate. Anaphase begins with the degradation of proteins that hold sister chromatids together, freeing individual chromosomes. The free chromosomes are then pulled by their kinetochores to opposite poles. At telophase, a cleavage furrow forms in the center of the cell. This indentation is made from a constricting belt of actin filaments surrounding the inside of the cell's circumference. Chromosomes cluster at opposite poles and begin decondensing as the nuclear envelope reforms around them. The spindle apparatus disassembles as the microtubules are broken down into tubulin monomers that can be used to form the cytoskeleton of the daughter cells. In animal cells, cytokinesis completes cell division by extending the cleavage furrow to completely separate the newly formed daughter cells. Since plant cell walls cannot be constricted by actin fibers, vesicles form an expanding membrane partition called the cell plate. Like animal cells, plant cells use cytokinesis to finish the division of the contents of the cytoplasm between the two identical daughter cells. During the cell cycle, certain checkpoints are encountered to make sure the process is occurring accurately. And if it is not, the cell cycle will stop at the checkpoint and correct, or possibly inhibit that cell from dividing. The first checkpoint is the G1S checkpoint, and is considered the primary point at which the cell cycle continues or stops. External signals and growth factors can influence the cell cycle and affect the progress at or before this critical checkpoint. The G2M checkpoint allows cells that have successfully completed all three phases of interphase to begin mitosis. The last checkpoint is the spindle checkpoint, ensuring that all chromosomes have attached to the spindle in preparation for anaphase. Growth factors, the size of the cell, and the nutritional state of the cell are all contributing factors in cell cycle regulation, ensuring that only certain cells divide at appropriate times. Once all the checkpoints in interphase are cleared, mitosis can occur. From interphase to cytokinesis, the entire process of cell division can take on average 10 to 20 hours in a typical plant or animal cell. Depending on the nature and use of the cell, the process can happen at different frequencies as well. In humans, our skin cells have a high turnover rate due to wear and tear, and go through mitosis very frequently, whereas other cells, such as adult neurons and muscle cells, rarely divide. The accuracy of mitosis, as well as the consistency of the checkpoints during interphase, ensure that most cells in a eukaryotic organism can produce identical copies of themselves. This process allows for growth and repair to prolong overall physiology as well as life itself.
Firstly, what is interphase? So it is the time interval between the end of one mitotic division to the start of the next one. So ये है time period end of one mitotic division to the start of next one. So this time period is known as interphase. So it starts from G1 and ends up into the G2 phase, that is gap 2 phase. Interphase is a period of intense biosynthetic activity, और जो cells हैं they are metabolically active. यहाँ पे जो भी basic requirements है वो पूरी की जाती है, in which the cell doubles in size increase हो जाएगा and duplicates precisely its chromosome complement. So ये सारे events इस phase में होते हैं, that is interphase. Then cell prepare themselves for the next division during this period. Basically cell खुद को prepare करता है for next cell division, and this interphase is further divided into 3 stages. First one is the G1 phase, that is first gap phase. Then S phase, the DNA synthesis phase. Here DNA replication occurs. Then comes the G2 phase, that is the second gap phase. Firstly, gap 1 phase or G1 phase. It is the period between the end of telophase and just before the entry of S phase is known as G1 phase. So जो telophase के पहले वाला time period है, after end of telophase and start of the S phase. S phase से पहले वाला और telophase के बाद वाला जो period है, that period is known as G1 phase or gap 1 phase. It is longest and more variable period of the cell cycle. Cell grows in this phase, metabolically active होते हैं cells and prepare itself for the DNA synthesis, और DNA की synthesis के लिए जो भी requirements चाहिए होती हैं, that is it involves synthesis of RNAs and proteins, and then mitochondria, chloroplast, endoplasmic reticulum, lysosomes, Golgi apparatus, vacuoles and vesicles are also produced. इस phase में ये सारे cell organelles भी prepare किए जाते हैं. The enzymes and substrates which are required for DNA replication or DNA synthesis during S phase are also synthesized during this phase. So इस phase में ये important events होते हैं. As a whole, cellular metabolic rate is very high in G1 phase. As a result, cell growth occurs. Cell जो है growth ज़्यादा करता है and it approximately doubles its size. So there are some checkpoints which are known as molecular checkpoints which are present at the end of G1 phase and allows or prevent the start of next phase. So G1 के end में यहाँ पे कुछ molecular markers और checkpoints present होते हैं जो ये check करते हैं कि जो G1 में जो भी requirements थी वो complete हुई है. The synthesis of various proteins, कोई इसमें aberration तो नहीं है, कोई इसमें disparity तो नहीं है. After that, after checking this G1 phase, this molecular marker or we can say checkpoint will allow the entry of the cell into the next phase, that is the S phase, which involves DNA replication. So the rapidly dividing cells, that is embryonic cells, lacks G1 and G2 phase, and it consists of very short S phase. जो embryonic cells होते हैं, zygote से पूरा जो embryo बनता है, and after that it will form the adult human being or adult eukaryotic organism. So that lacks the G1 and G2 phase, और इसमें जो S phase होता है बहुत ही short होता है. Typical human proliferating cell with a total cycle time of 24 hours. Normally जो human cell की बात करते हैं, so इसका जो time period होता है cell cycle का, that is approximately 24 hours. इसमें जो G1 phase होता है, that lasts for approximately 11 hours, S phase for 8 hours, G2 phase about for 4 hours, and M phase is about 1 hours of duration. So ये time period है. Total 24 hours का time duration होता है जो normal proliferating eukaryotic cell है, human cell, and it is further divided into different phases. That is G1 phase consists of 11 hours, S phase of 8 hours, G2 phase of 4 hours, and the mitotic phase is of 1 hour. The budding yeast cell is only the budding yeast cell cycle is only 90 minutes. जो budding yeast cell cycle है, yeast का cell cycle है, वो सिर्फ 90 minutes का होता है. Shorter cell cycle, that is 30 minutes or less, occurs in early embryonic cell because it lacks G1 and G2 phases. Next comes G0 state. Some cells in adult animals ceases cell division. For example, we have some cells like we have neurons, striated muscle cells, skin fibroblast, and liver cells. ये जो cells हैं, they will not enter into the S phase, and they will go into another phase that is known as G0 phase or quiescent stage. यहाँ पे जो cell है ये metabolically active रहते हैं, but they are unable to divide. But under certain conditions, if they are required, they will undergo the cell division. So this stage, when the cell ceases, the cell division, or it will not enter into the S phase, is known as G0 stage. These cells exit G1 phase or gap 1 phase to enter a quiescent stage or inactive stage of the cell cycle, which is known as G0 stage, where the cell remains metabolically active but no longer proliferate or divide. यहाँ पे जो सेल है metabolically active रहेगा but divide नहीं करेगा. These arrested cells can be induced to divide again under certain conditions. For example, liver cells normally neither grow nor divide, but liver damage rapidly induces them to divide. Under these conditions, these liver cells can divide. Next comes synthetic phase or S phase. Here, the DNA replication occurs. So S phase is intermediate phase between G1 and G2 phases, that is gap 1 or gap 2 phases, and DNA synthesis occur in this phase. So this is very important event which occur during this S phase. It is a highly specialized phase of interphase. ये सबसे ज़्यादा specialized phase है interphase का. Before a cell can divide, it must produce a new copy of its chromosome. This is very important event which occurs during this phase. Animal cells in G1 phase are diploid. जो एनिमल सेल्स हैं G1 फेज में डिप्लॉयड होते हैं. फॉर मेकिंग अ न्यू कॉपी ऑफ क्रोमोसोम इट नीड्स रेप्लिकेशन. एट द एंड each chromosome has been copied to 2 complete chromatids which remain joined together at the centromere until M phase that soon follows. तो जो एंड है एस फेज का, उसके एंड में जो क्रोमोसोम का अमाउंट है वो डबल हो जाता है. मीन्स इसमें जो जेनेटिक कंटेंट है वो डबल हो जाता है. क्रोमोसोम नंबर रिमेन सेम, बट जो सिंगल आर्म है क्रोमोसोम की दैट कंटेन्स द material of a full chromosome. During this phase, DNA content of the cell increases from 2n to 4n, that is diploid to tetraploid, but the number remains same. DNA content remains at 4n, that is tetraploid for the cell in G2 and M phase, decreasing to 2n after the cytokinesis. जो cytokinesis के बाद cytokinesis होता है उसमें ये क्रोमोसोम नंबर फिर से क्रोमोसोम का अमाउंट फिर से डिप्लॉयड हो जाता है. और after following cytokinesis, there also occur a very important event, that is histone synthesis and new centromeres in animal cells, which are formed during this phase, that is S phase, for the mitotic spindles, which helps in mitotic spindle formation. So synthesis of these histone proteins and centromeres, and it occurs simultaneously during the replication of DNA. Experimentally, DNA content can be determined by डिवाइस जिसका नाम है फ्लो साइटोमीटर. अगर हमने डीएनए का कंटेंट मेजर करना है उसके लिए हम इस डिवाइस को यूज़ करते हैं, विच इज़ फ्लो साइटोमीटर. सो आफ्टर एग्जिटिंग द G1 फेज, द सेल एंटर इनटू द S फेज, और इसमें जो इंपॉर्टेंट इवेंट होता है दैट इज डीएनए रेप्लिकेशन. डीएनए की सिंथेसिस होती है, एंड अलॉन्ग विद डीएनए रेप्लिकेशन देयर अकर्स द सिंथेसिस ऑफ हिस्टोन एंड सेंट्रोमियर. नेक्स्ट कम्स गैप 2 और G2 फेज. It is the period from the end of S phase until the mitosis is started is called G2 phase. So M phase से पहले और S phase के बाद का जो time period है, that phase is known as G2 phase or gap 2 phase. It is the shortest part of interphase, shortest duration का होता है interphase में. अगर देखें, in this phase intensive cellular synthesis occur, mitochondria and chloroplast divide. इस phase में ये division होता है इनका. RNA and protein synthesis remain continuous. Energy stored increases. Cell increases in size, and centrosomes of animal cells are positioned at opposite locations of the cytoplasm. इसमें जो सेल में centrosomes हैं वो दोनों ही opposite poles की तरफ अरेंज करना शुरू करता है. That helps in formation of esterase and spindle fibers. Mitotic spindle begins to form. In the interphase, there are 2 control points such as G1 to S checkpoint and G2 to M checkpoint, at which the cell takes a decision on whether to proceed or not to at the next step. So there are 2 checkpoints. First is between G1 and S phase. So ये जो checkpoint है, second is between G2 and M phase. जो ये checkpoints हैं, ये check करते हैं कि सेल में जो भी चीज़ें चाहिए थी, requirements हैं सेल डिवीजन के लिए, वो सारी complete है. If they are complete, then it will allow the cell to enter into the M phase. Otherwise, it will stop the cell before entering into the M phase. So 2 control points are also called checkpoints. इन control points को checkpoints भी कहते हैं. End of G2 phase is mediated by cyclin dependent kinases, that is abbreviated as CDK. जो एंड है G2 फेज का ये मेडिएट और इसमें हेल्प करता है साइक्लिन डिपेंडेंट काइनेज एंड बाय द B1 साइक्लिन. सो दीज आर द प्रोटीन्स और ये जो CDKs एंड साइक्लिन प्रोटीन्स है दे विल फॉर्म द चेक पॉइंट. ये अलाउ करेगा इसकी एंट्री को इनटू द M phase आफ्टर पासिंग द G2 phase. This CDK1 and B1 complex acts as a checkpoint and activated starting of M phase और ये help करते हैं activation of this starting of M phase at mitotic phase, which involves division of nucleus and division of cytoplasm. Next comes the mitotic phase. This word mitosis is derived from Greek word mitos, which means thread. It is the most complex phase. And the term mitosis was coined by Walter Fleming in 1882. August Weismann in 1887 used the term equational division for mitosis because here the chromosome number remains same in parent cell as well as in the daughter cells. The M phase leads to separation of replicated DNA into two daughter nuclei without recombination. The daughter nuclei have the same chromosome combination as that of the parent nuclei. And mitosis is of two types. One is open mitosis and other is a closed one. Open means when it occurs outside the nucleus. It means the nuclear membrane है वो disappear हो जाती है. Closed means when the mitosis occur within the nucleus without disappearing of nuclear membrane. The M phase consists of four stages, namely prophase, metaphase, anaphase, and telophase. Then it is followed by cytokinesis, which means division of cytoplasm. And M phase ends up with two new cells. So first phase of this mitosis is prophase. Pro means before, phases means appearance. This prophase starts immediately after the G2 stage of interphase, and here the chromosome looks like thin thread and uncoiled in the early prophase but becomes shortened, coiled, and more distinct during the mid prophase by the process of condensation. And this condensation is due to dehydration and coiling of the chromatin fibers. जो chromatin fibers हैं वो coil होते हैं और dehydration का process होता है जिससे जो chromatin fibers है they coil around each other to form a thick rod-like structure जिन्हें chromosomes कहते हैं, which appear at the end of prophase, that is late prophase stage. So condensation occurs due to dehydration and coiling. In the late prophase, chromosomes appears more conspicuous, short, thick, and longitudinally double. So late prophase में हम chromosomes को देख सकते हैं. The two chromatids of each chromosome held at point known as centromere or primary constriction. जो क्रोमोसोम हैं जो उनकी क्रोमेटिड्स हैं वो सेंटर में एक पॉइंट पे जुड़ती हैं और उस पॉइंट को बोलते हैं सेंट्रोमियर और प्राइमरी कंस्ट्रिक्शन. बटन लाइक स्ट्रक्चर नॉन एज काइनेटोकोर एट द आउटर सरफेस ऑफ सेंट्रोमियर. इसके बाहर एक बटन लाइक स्ट्रक्चर होता है सेंट्रोमियर के बाहर जिसे काइनेटोकोर कहते हैं एंड इट हेल्प्स इन बाइंडिंग विद स्पिंडल फाइबर्स. स्पिंडल फाइबर्स यहाँ पे जुड़ते हैं काइनेटोकोर में. सो दिस इज द real image under microscope और ये जो stage है this shows the late prophase, this is the early prophase और यहाँ पे जो chromosomes है condense होकर rod like structure की तरह में visible होते हैं. Next this stage is metaphase stage and this is anaphase, telophase and finally the cytokinesis. Then if we talk about chromosome, so chromosome consists of two identical arms which are known as chromatids. इन्हें क्रोमेटिड्स कहते हैं और सेंटर में ये जुड़े होते हैं एक डिस्क लाइक स्ट्रक्चर जिसे बोलते हैं सेंट्रोमियर. इसकी जो शॉर्ट आर्म्स होती है क्रोमोसोम्स की विच आर नोन एज पी आर्म्स. देन द लॉन्गर आर्म इज नोन एज क्यू आर्म एंड इट इज़ फॉर्म्ड ऑफ डीएनए मॉलिक्यूल. जो डीएनए मॉलिक्यूल्स वो कॉइल्ड होंगे बाय द प्रोसेस ऑफ सुपर कॉइलिंग एंड दे विल फॉर्म दिस रॉड लाइक स्ट्रक्चर जिसे क्रोमोसोम कहते हैं. और इसके बाहर और डिस्क लाइक स्ट्रक्चर होता है जिसे in animal cell and some lower plant cell, the spindle fibers are formed radiating in an outward direction from the two centrioles called ester. जो animal cells हैं उनमें बाहर की तरफ को कुछ rays like structure निकलती हैं जिन्हें कहते हैं esters और इस तरह के mitosis को बोलते हैं estral mitosis. Here the centrioles are missing in plant cells जो प्लांट सेल्स होते हैं उनमें सेंट्रियोल्स एब्सेंट होते हैं और उसमें जो माइटोसिस होती है दैट इज नॉन एनेस्ट्रल माइटोसिस, मींस इसमें एस्टर रेज नहीं बनती है और इस तरह के माइटोसिस को बोलते हैं एनेस्ट्रल माइटोसिस। स्पिंडल फाइबर्स आर फॉर्म्ड बाय एग्रीगेशन ऑफ माइक्रोट्यूब्यूल्स। जो स्पिंडल फाइबर्स हैं वो माइक्रोट्यूब्यूल्स के एग्रीगेशन से बनते हैं एंड दीज माइक्रोट्यूब्यूल्स आर मेड अप ऑफ प्रोटीन जिसे कहते हैं ट्यूबुलिन प्रोटीन। द न्यूक्लियोलस बिकम्स स्मॉलर इन साइज ड्यूरिंग लेट प्रोफेज। जो न्यूक्लियोलस है वो स्मॉलर साइज का हो जाता है और न्यूक्लियर मेम्ब्रेन भी स्टार्ट होती है डिसअपियर होना एंड द न्यूक्लियर मेम्ब्रेन एंड न्यूक्लियोलस डिसअपियर एट द एंड ऑफ प्रोफेज। जो लेट प्रोफेज है वहाँ पे न्यूक्लियोलस और न्यूक्लियर मेम्ब्रेन दोनों ही डिसअपियर हो जाते हैं एंड अमंग दीज ऑल द फेज ऑफ माइटोटिक फेज, जो प्रोफेज है इट इज़ द लॉन्गेस्ट फेज ऑफ माइटोसिस। नेक्स्ट कम्स मेटाफेज। मेटा मीन्स आफ्टर, फेज मीन्स अपीयरेंस। दिस फेज बिगिंस आफ्टर प्रोफेज। प्रोफेज के बाद ये फेज स्टार्ट होता है। इट इन्वॉल्व्स मिक्सिंग ऑफ मैट्रिक्स ऑफ विद साइटोप्लाज्म। जो न्यूक्लियर मटेरियल है न्यूक्लियर प्लाज्म दैट मिक्स विद द साइटोप्लाज्म। देन द क्रोमोसोम्स आर मूव टूवर्ड्स द मिडल पोर्शन, जिस प्रोसेस को बोलते हैं कॉन्ग्रेशन। सो दिस इज द प्रोसेस बाय विच द क्रोमोसोम्स अरेंज देमसेल्व्स इन द मेटाफेज। प्लेट और इक्वेटोरियल प्लेट सेंटर में क्रोमोसोम्स अरेंज करते हैं और इस प्रोसेस को बोलते हैं कॉन्ग्रेशन। Force is generated by the motor proteins and arms of chromosomes. Chromosomes और इसी phase को बोलते हैं pro-metaphase. Then chromosomes are oriented in the middle of spindle between the two poles at equatorial plate और metaphase plate. So chromosomes lie करते हैं इस plate पे जिसे बोलते हैं equatorial plate और metaphase plate और chromosomes are clearly visible in this phase जिसे वो बोलते हैं metaphase. So in this phase we can observe, we can count the chromosomes. Sister chromatins of each chromosomes are joined together at a point जिसे बोलते हैं centromere, but their arms are free that we have already discussed in previous slide. Two kinds of spindle fibers are formed. Some are long, extending from one pole to the another. So some fibers are long जो extend करते हैं एक पोल से दूसरे पोल में and some are और इन्हें कहा जाता है continuous fibers भी and some fibers gets attached to the chromosome at the centromere जिन्हें बोलते हैं chromosomal fibers. So दो तरह के fibers हैं, long fibers or continuous fibers. Second one are the chromosomal fibers. So this is the original picture which shows the metaphase. This is the cell और इसके center में present हैं chromosomes in the equatorial. Next comes the anaphase. Ana means up, phases means arrangement. Here chromatids separate from each other at the centromere and is called daughter chromosome. यहाँ पे जो chromosomes हैं वो center से centromere के breakdown से दो parts में divide हो जाते हैं. Here the chromosome breaks at centromere and the sister chromatid moves to the opposite end that we can see in this animated photograph. So जो arms हैं कि arms ये opposite pole में move करती हैं. Daughter chromosomes moves to the opposite poles of the spindle. Daughter chromosomes appear V, U or J shaped during their movement towards the poles. And anaphase changes into the next stage called telophase. This slide shows the various shapes of chromosome on the basis of presence of centromere. So firstly centromere location at the middle, so that type of chromosomes are known as metacentric chromosome. इसमें जो centromere है इसकी position होती है center में and when during anaphase when the chromosomes moves, so the appearance of this chromosome is V shaped. V shape में appear होते हैं. Next when the chromosomes are present between middle and the end, so that type of chromosome is known as submetacentric, means center से थोड़ा सा ऊपर की तरफ present होता है centromere and the chromosomes appear like L shaped. Next close to the end इस तरह के chromosome को बोलते हैं acrocentric where it is subterminal in position and जो इसकी chromosome की appearance है during anaphase it is J shaped. Sometime the centromere is present at the tip और ऐसे chromosomes को बोलते हैं telocentric chromosomes and these chromosomes appear like. Next comes the final stage that is telophase. Here two daughter groups of chromosomes reach to their respective poles. जो daughter chromosomes हैं वो अपने अपने poles में पहुंचते हैं. Here there is reconstruction of daughter nucleus. जो nucleus है वो फिर से reconstruct होता है, means nuclear membrane फिर से बनती है, nucleolus भी फिर से reappear होता है. Nuclear membrane begins to form around each group of chromosome. Nuclear membrane फिर से बनती है and each chromosome becomes hydrated. जो प्रोफेज के एंड में हमने पढ़ा था कंडेंसेशन, यहाँ पे डीकंडेंसेशन हो जाएगी, मीन्स रिवर्स ऑफ कंडेंसेशन बाय द प्रोसेस ऑफ हाइड्रेशन एंड अनकॉइलिंग. जिससे chromatin fibers बन जाएंगे thin fibers. The nucleus again reappears in each daughter nucleus, and the spindle fibers disappear. So these are the events which occur during the last phase of mitosis. Here, nuclear membrane reforms, nuclei reappear, chromosomes unwind into chromatin fibers. There are proteins which are known as myosin 2 and actin filaments ring contact to cleave the cell into two. And this is the animal cell, or animal cell में जो cell division होता है, that is division of cytoplasm. It is the process of formation of a furrow which is known as cleavage furrow. This division of nucleus is followed by division of cytoplasm, that is known as cytokinesis. Cytokinesis is the division of cytoplasm. और जो animal cells हैं उनमें division of cytoplasm it occurs by the process of formation of cleavage furrow, जो कि बाहर से अंदर की तरफ को बनता है towards center. While in case of plant जो इसमें cell division होता है means cytoplasm का division होता है that starts from the center, जिसे बोलते हैं cell plate formation method. So the division of nucleus is also known as karyokinesis. Nucleus के division को karyokinesis बोलते हैं. It is followed by division of cytoplasm which is known as cytokinesis. And the division of cytoplasm into two daughter cells may takes place in two ways. दो तरह से होता है. In plants, the cell division of this cytoplasm division takes place due to the formation of cell plate that I have already discussed, where the formation of this plate begins at the center. So ये start होता है center से, which moves towards the periphery in both the side, dividing the cytoplasm into two daughter cells. While in case of animal cells, the separation of cytoplasm starts by the furrowing of plasmalemma or plasma membrane, which is known as cleavage furrow. और इसे बोलते हैं cleavage furrow. ये start होता है outer side से toward the center and delimits the cell and separates the two cells, daughter cells. This results in division of cytoplasm into two daughter cells. Next comes significance of mitosis. Firstly, genetic stability. It helps in preserving and maintaining genetic stability of the particular population. It also helps in splitting of chromosomes during cell division and generates two cells which are identical to their parents. Next, repairing of wounds. जो injured part है repairs wounds or broken part of the organisms rapidly by producing new cells. Next, it also helps in asexual reproduction. It is used in production of genetically similar offsprings which are known as clones. For example, in case of yeast, there is budding in yeast and hydra, then binary fusion in amoeba. So these are because of asexual reproduction, and the division occurs with the help of mitosis. Keeping the chromosome number constant in all body cells of an organism. So mitosis helps in maintaining the chromosome number in all the cells, and is responsible for the development of zygote into adult. So यही division है mitosis which helps in formation of adult from the zygote. It also helps in maintaining qualitative stability. Mitosis maintains shape and size of cell remain constant by this division, and sometimes it also leads to some diseases like tumor, cancer, etc. When there is uncontrolled mitosis or uncontrolled cell division. So these are the various significance of mitosis. So this was all about our today's discussion about cell cycle and mitosis. We hope you will get some idea from this presentation. If you have any questions, queries, and any suggestions, you can give it in the comment section. Thanks for watching. Have a great day.